Namaste everyone, I'm Dr. Ankita Sawant. Welcome to Physio Vitals. And today I'm back with the third series of embryonic development of nervous system. So today's lecture will be on how the head and brain is developed. So this is the third part of the continuation series. So I'll be providing the link of first two lectures as well, so that we all will be on the same page. So we saw neural tube, how the neural tube is developed. Now we'll see how the brain and then we saw how the spinal cord has been developed. Now further we are going to see how head and brain is getting developed. So once the neural tube enlarges at the cephalic end is head end. So once the neural tube enlarges around 3 to 4 week, this step occurs. The cephalic portion of neural tube enlarges into three regions, and that three regions are primary brain vesicles, which is prosencephalon, which is the forebrain, mesencephalon midbrain, and rhombencephalon, which is the hindbrain. So neural tube enlarges by 3 to 4 week at the cephalic end into three regions: primary brain vesicles, prosencephalon forebrain, mesencephalon midbrain, rhombencephalon hindbrain. Now, there is a fissure between prosencephalon and between prosencephalon and mesencephalon. Important in human for bipedalism. Here, the brain is at 90 degree from the spinal cord. Alright. Now, by 4 to 5 weeks, these primary brain vesicle further develop. Like prosencephalon develops into telencephalon, which in future becomes cerebral hemisphere. Just remember cerebral hemisphere, TC. Then diencephalon into thalamus and hypothalamus, DTH. Remember mnemonic wise, it will be easy to memorize. Then mesencephalon is just the midbrain. Rhombencephalon further develops into metencephalon, which is pons and medulla, MPC, and myelencephalon, which is for the medulla, MM. So telencephalon into cerebral hemisphere, diencephalon into thalamus and hypothalamus, mesencephalon as it is midbrain, rhombencephalon into metencephalon, which is pons cerebellum, myelencephalon is future medulla. Now, brain formation. At around 11 to 13 weeks, there is massive proliferation of neuroblasts in cephalic neural tube. By 11 to 13 week, further massive proliferation occurs of neuroblast. So in cephalic neural tube, causing folding due to lack of space within the cranium. Alright. So as you can see here, midbrain, this is the cerebrum, this is the cerebellum, this is spinal cord, medulla. This is the 13th week. How it gets, like from here, how it slowly develops, and then it gets congested, folds and takes a nice shape. Pharyngeal arches and cranial nerves. Pharyngeal arches, similar to somites in the lower part of embryo. Each pharyngeal arch consists of ectoderm tissue, cranial nerve, which is cranial nerves and skin of face. Phalangeal arches, which are similar to somites in the lower part of the embryo. Likewise, each pharyngeal arch consists of ectoderm, which is cranial nerve and face, skin of the face. Mesenchyme, mesodermal tissues, which are the musculature of face and neck. Endoderm tissue, phalangeal epithelium. So important note here is this results in segmental development of head and neck, similar to somites. So in this diagram, as you can see, endoderm, this is ectoderm, this is the phalangeal muscle. Here we have the brachial nerve. Here we have the cartilage. This is the artery, phalangeal arc, this is phalangeal pouch. Next is the formation of ventricles, very important. The neurocele of neural tube becomes the ventricle of adult brain. Neurocele of the neurotube becomes ventricles of the adult brain. So we have lateral ventricle, third and fourth ventricle. In lateral ventricle, we have two ventricles, 1 and 2. So it sits in the cerebral hemisphere, telencephalon. This is shaped due to folding of the brain during the development, which consists of a frontal or anterior horn. It has body, occipital or the posterior horn, and a temporal or the inferior horn. So we have anterior, body, posterior, inferior horns. Third ventricle sits in the diencephalon. Lateral walls are formed by thalamus and hypothalamus. Connects with the fourth ventricle via cerebral aqueduct. Next is the fourth ventricle, which sits in the brainstem and it's continuous with the spinal cord canal. So here we can see this is the ectoderm, neural crest, this is the neural tube, and this is the somae. Here further development happens. From this we have forebrain, midbrain, hindbrain. Now here it further gets developed into the two lateral ventricles. This is the third ventricle. Here we have the fourth ventricle. Between third and fourth ventricle we have cerebral aqueduct. Now further in the adult brain, this is the lateral ventricle 1 and 2. Here we have the third ventricle. This is cerebral aqueduct and rhomboid shaped fourth ventricle. So these are the ventricular system. So this is the how it is formed. The neurocele of the neural tube becomes ventricle of the adult brain. Got it? So that was till this series of how the embryonic development occurs of the nervous system. So we saw how neural tube is developed, what is mesoderm, what is neurulation, what is blastocyst, neurulation, then neural tube is formed. From there, how neural tube gets into spinal cord formation. From there, how brain and head and brain is formed. Then we saw how ventricles are formed. So that was till this part of the lecture. Further, I'll be covering the overview, organization of nervous system. So if you like the video, comment, and if you want this PDF, I'll be providing, just comment I want PDF and I'll provide you the link. So stay tuned for further upcoming amazing videos. Thank you very much. Keep learning.